And finally, Jenna Calvert, who's a postdoc with Bob Schnabel in the Animal Genomes Group at the University of Missouri. So I will let them present their work today, and we're going to have all three talks and then a period for discussion after the talks at the end. I will stop sharing and let them begin. Thanks.
Hi everyone, hopefully you can see my screen. My name is Yana Stefanova, and today I will tell you about immunogenomics, and we are focusing on analysis of adaptive immune systems in ruminant species. And with the genomic approach, we are targeting adaptive immune loci. In most mammalian species, there are 7 adaptive immune loci, 3 immunoglobulin loci, IgH, IgK, and IgL, and 4 T-cell receptor loci. TR alpha delta actually located together in the same position, TR beta and TR gamma. They encode immunoglobulins or antibodies and T-cell receptors respectively, and they together are considered to be adaptive immune agents. The main feature of these agents is that they are not encoded in this loci directly. Instead, they present results of somatic genomic recombination. And this recombination process affects germline immunoglobulin or T-cell receptor genes located in corresponding locus. And we distinguish 3 types of these genes, V or variable, D or diversity, and J or joining. The recombination process randomly selects 1 gene from each family V, D, and J, and concatenates them together, thus forming a VDJ sequence. And in the chain of corresponding adaptive immune receptor, that VDJ sequence would be responsible for binding an antigen. So the diversity of VDJ sequencing is a crucial part of generating the primary diversity of adaptive immune receptors, and there are various selection mechanisms that identify important adaptive immune receptors and multiply them in copies. In antibodies, additional process improving binding features of antibody is also called somatic hypermutagenesis. This process randomly changes individual bases in the VDJ sequence, thus hoping to generate antibodies with better binding and neutralization processes. So together it forms a rapidly evolving system that is highly individual, highly specific to a subject, so it's really challenging to compare even subjects of the same population. However, we know that the system is important to studying susceptibility to diseases, developing better vaccines, and understanding how pathogens and host organisms coexist together and co-evolve. From studying mammalian immunoglobulin and T-cell receptor loci, we know that they are extremely diverse even in the germline form. If you look at, for example, immunoglobulin heavy chain locus in carnivore order and in rodent order, we basically won't see any similarity. And the similarity is even sometimes not observed for species from the same order, suggesting that these loci evolve very, very rapidly. Immunoglobulin loci have highest divergence compared to T-cell receptor loci, but even for T-cell receptor loci, we see that 2 arbitrarily selected species share only 10% similarity, which is very low. And it means that to actually study this fast process of evolution, we need to look closer at closely related species. So the Ruminant T2T project is a fantastic opportunity for us to trace events of evolutionary development of this rapidly evolving loci at a very small level. And with looking at closely related species, we can see at what point they start to diverge, so we don't see any similarity. Here you can see pairwise dot plots showing alignments of immunoglobulin heavy chain loci for several ruminant species, and also to whales just for comparison. So we already don't see any similarity between whales and ruminant species. But if you look closer, for example, we compare the cattle and the yak, we see that they actually share repeat copies, even though the layout of this repeat copies is different in these both species. It's also the same for the oryx. However, the repeat copies get shorter. And if you move to the deers or giraffes, then the similarity already starts to disappear. So we actually can narrow down at what point we can still see the similarity and at what point we don't see it at all, and what kind of sequence features lead to this diversity. So this is a huge work in progress, and right now we are acquiring the data. However, it already shows some preliminary results, allowing us to build comparative studies. Additional angle that also makes the ruminant clade interesting is an alternative mechanism of antibody diversification found specifically in ruminants, and for now in the literature it's only reported for cattle. This process describes additional process of antibody diversification that relies heavily on generation of cysteines in one of antigen binding site called CDR3. In many antibodies, this antigen binding site is just a loop, but when we have cysteines there, they start forming disulfide bonds and thus change the structure of a loop into more complicated structural shapes. It turned out that cattle has a special D gene called IGHD82, and it has 4 cysteines in the germline form, but also multiple templates for cysteine generation using somatic hypermutations. So there is a very high chance that a randomly generated single nucleotide change creates a new cysteine. And as a result, the antibody will likely change the layout of cysteine bonds and will have a different structure. So here, instead of having highly diverse VDJ recombination sequences, we can generate highly diverse antibody structures using the same gene over and over. And that seems to be a mechanism that probably compensates for something else. So an interesting direction would be to actually find out what is this compensation effect and what its evolutionary origin. These antibodies are also interesting as possible antibody drugs. It's already shown that they are efficient neutralizers of several human viruses such as HIV. So there are additional efforts actually right now outside of our project to humanize them and make them efficient drugs. So it also shows that we probably should look closer into ruminant species to see maybe we can identify additional drug candidates. To study expressed adaptive immune repertoires, we actually need to go into tissues that have them, and usually this is either blood or spleen tissues. And in the Ruminant T2T project, we mostly work with blood. 2 common ways of sequencing expressed VDJ sequences include targeted repertoire sequencing or untargeted ISO-seq sequencing, and we actually had a lot of luck with ISO-seq Kinnex because it's first of all unbiased, so we can sequence all adaptive immune receptors at once without creating specific primers, but also we can cover the full transcript with a single PacBio hi-fi read and have a really nice quality. We can see that proportions of individual adaptive immune chains is a variable characteristic itself. And we can see that from one species to another species, these proportions might change significantly. For example, the cattle is a champion in production of IgL and also TRR TRA, while for ELK we see a significant portion of IGH chains. So probably that might be another basis for disease-specific studies, because some vaccines actually applied for all of the species, and they expect to target specific types of chains. So when we see that some chains are underrepresented, it could mean that vaccines are not that efficient for the species as compared to other species. So that's currently our tool of choice for studying expressed adaptive immune repertoires in ruminant species. And we applied it to several species from the ruminant phylogenetic tree. And first of all, we looked at their immunoglobulin loci in the germline genome. From the previous slides, we already seen that they are diverse. However, we also see that they are repetitive. And the majority of V genes are concentrated in this nearly tandem or mosaic structures, meaning that the diversity of V genes is very low because they located in copies of repeats. And we decided to use this information as another dimension for the comparative study of ultra-long antibodies. So we scanned immunoglobulin heavy chain loci of various species, and we noticed that ultra-long D genes possibly producing ultra-long antibodies are also found in some deers and giraffes. However, they are missing in some other species, which creates an interesting story, meaning that some species acquire these mechanisms and some others don't. We compared specifics of these genes, and they turn out to be very similar. They all have opportunities to create additional cysteines, and we checked expressed antibody repertoires, and we indeed see that the distribution of lengths of antibodies is bimodal in species with germline long D genes. So it means that these D genes are not sitting in the genome, they also produce antibodies that can be functional. The lengths of ultra-long antibodies is slightly different from species to species, and this can be explained by differences in ultra-long D gene lengths. And sometimes we see that these genes can be present in multiple copies, so that's probably another source of the diversity. But nevertheless, right now we can extend the existing understanding and say that ultra-long antibodies are not just a feature of cattle and cattle-like animals, it's a feature of the ruminant clade. However, there is a question, does it mean that the ancestral ruminant species also had ultra-long antibodies, and then some species lost these antibodies, for example, ones shown here, or these ultra-long antibodies emerged independently in the species? And I think that the first hypothesis is right, because we also looked at the positions in the genome where these ultra-long D genes are located, and they are located at homologous positions, meaning that probably the ancestral species also had them, and for some reason these D genes accumulated deleterious mutations in some other species. And when we compared the presence of these ultra-long D genes with repetitiveness of IGH locus, it also gave us additional ideas why this happened. It turned out that these two characteristics are positively correlated, and it means that when we have low diversity of germline immunoglobulin genes located in highly repetitive loci, we usually observe that these ultra-long antibodies are present in the species, and they probably compensate for loss of diversity of V genes and thus VDJ recombinations. However, when the repetitiveness is low and the diversity of V genes is higher, antibody repertoires can create this diversity just by VDJ recombination process, the very basic one. And in this case, we probably don't have this pressure to develop alternative mechanisms, and ultra-long D genes just don't have enough selective pressure. So this is a comparative study that we're currently working on, but also ultra-long antibodies have applications in studies of cattle diseases and probably deer and giraffe diseases. And previously, we observed that the fraction of ultra-long antibodies goes high with vaccination against the bovine respiratory disease, and this fraction even positively correlates with antibody titers against bovine respiratory disease. However, we see that this correlation is far from perfect, meaning that there are probably additional factors contributing to this variance. One of these factors could be a different number of copies of repeats in cattle IgH loci. It's not something completely unexpected. When we have mosaic repeats, it means that probably they are subject to unequal crossover, and from subject to subject, we can start observing different number of copies. And it means that some genes might be present in more copies than others. Indeed, if we look at existing cattle genomes, we see that this variance is indeed huge, and locus of IgH loci can go from 400 kilobases to a megabase, and of course they affect the number of genes they encode. It also opens a challenge that we're trying to solve right now, and it's developing good nomenclatures for these genes. The existing nomenclature assigns names of Ig genes according to their position in the locus, and it doesn't take into account that some of these genes are actually copies of each other. And it also is not robust to new genomes that can have different number of copies and differently, and maybe some other genes that we don't observe in the reference one. So analysis of, again, available cattle assemblies show us that indeed some of these genes are located in clusters representing extremely high similarity, more than 99%, and actually these gene names should be changed into something identical. And there is also a huge and very populated cluster combining several gene names, and it's an open question: should we break it or should we keep it as some really big cluster? But regardless of what we choose, it's going to affect the analysis of antibody repertoire specific to disease, because we will assign VDJ sequences to different genes and perform association studies differently. This problem is currently a priority for us, and we're trying to solve it in a transparent manner. We work with the International Union of Immunological Societies to make this approach versatile, because judging from the organization of immunoglobulin and T-cell receptor loci in ruminant species, I would say that this problem is actually applicable to other species, not just cattle. So we are very open to collaboration. So if you have some interesting case study, please feel free to contact me. We have a very nice group of people with different expertise from evolutionary side, genome assembly, and immunogenomics. And of course, I'm honored to work with Ruminant telomere-to-telomere project, because this is a great source of data and a fantastic group of scientists. And finally, I'd like to thank our PhD students, and especially Marie Paspello, who done a lot of work that I showed to you. And thank you so much for your attention.
Thanks, Yana. That was wonderful. We're going to hold off questions until the very end, but real interesting stuff. Next up, yes.
Yeah. So I hope you can see it full screen, right? Yeah. So hello everyone, my name is Inês Dias, and today I will be showing you we have been using Asian muntjac deers as model systems in our lab to understand how dividing cells adapt to karyotypic evolution. Wait. Okay, so the first species I will be talking about today is the Indian muntjac, which is the mammal with the lowest non-chromosome number, with a diploid karyotype comprising only 6 giant chromosomes. And not only these chromosomes are much bigger than human chromosomes, for example, but their centromeres are also much bigger than human centromeres, for example. With this exception case of the chromosome 3X that presents a giant compound centromere that has almost 2 micrometers of length. And so having this system implemented in our lab created this amazing opportunity to study mitosis in a way that was not possible before, as fewer chromosomes and bigger centromeres make it easier to visualize unknown mechanisms throughout the whole cell division process. And so when I tell you that the Indian muntjac centromeres are much bigger than human centromeres, for example, they really are. And so, as you probably know, a human centromere is able to bind more or less 20 microtubules. And in the Indian muntjac, even the smallest centromere binds 50% more of those microtubules, as you see here. And the largest centromere on chromosome 3X is actually able to bind 3 times the number of microtubules when compared to a human centromere. And as you can see here, not only the microtubule binding capacity is proportional to this kinetochore size in Indian muntjac, as well as the other functional layers of the kinetochore. So as you can see here, the largest kinetochore in Indian muntjac has more or less twice the volume as the smaller ones. And so this prompted Danica at the time to be able to ask questions that were not possible to ask before. And the first one was obviously how the kinetochore size affects chromosome segregation. And so we already know from human cells, as you can see here, that there are 2 main pathways for chromosome congression from the pulse to the metaphase plate before chromosomes are actually able to segregate into daughter cells. And these pathways are either SEMP-E dependent or SEMP-E independent. So SEMP-E is this motor protein that is able to bring kinetochores to the metaphase plate. And so when Danica inhibited SEMP-E motor protein in Indian muntjac cells, here we have a still where you have an arrow pointing to the biggest kinetochore in Indian muntjac cells. You can see that she was able to find 3 very different scenarios. The first one being that all chromosomes stayed at the pulse, so were not able to congress without SEMP-E. Then you also have cells that had no chromosomes at the pulse and cells with only a few chromosomes at the pulse. And so what this indicates is that any chromosome might use both pathways, so either SEMP-E dependent or SEMP-E independent to congress regardless of kinetochore size. The other thing that Danica noticed at the time is that smaller kinetochores, as you can see here, have more or less twice the probability to stay at the pulse if we inhibit SEMP-E when compared to large kinetochores in the Indian muntjac cells. And this is a strong indication that these chromosomes with larger kinetochores are actually less dependent on this motor protein to align. Lastly, Danica used this system to understand if kinetochore size would influence segregation fidelity. And for this, she started by doing a monostral treatment and washout. And what this does is basically promoting erroneous kinetochore microtubule attachments. And these erroneous attachments might be either scintillic, as you see here, where you have both sister chromatids of each chromosome binding to microtubules from the same pool, or merotelic attachments where you have the same sister chromatid attached to microtubules from both poles. And that's also why it creates this very distorted kinetochore that is feeling pulling forces from both sides of the spindle at the same time. And so the goal of this experiment was to understand if by enhancing this chance of erroneous kinetochore microtubule attachments to occur, if there would be a bias for the chromosome with the biggest kinetochore to lag behind during chromosome segregation, as you can see here. And again, what Anitsa saw was that indeed the chromosomes that had the largest kinetochores had a very strong bias to be the ones that will lag behind during chromosome segregation. And so all these results prompted Danitsa's model that I'm showing you here, and that also shows that chromosome alignment and segregation are biased by kinetochore size. So later, Anna Almeida joined our lab for a PhD study, and she was also using the Indian Moonchuck system. And at the time, Anna started a mitotic screen in these cells by depleting 63 conserved mitotic genes using siRNAs. And so after analyzing the effects of these depletions, as you can see here, in mitosis, she then scored the severity of mitotic effects of each depletion. And she saw that AUS6 or augmin depletion was causing the most severe mitotic effects in Indian Moonchuck cells. And so then she followed up with this hit. And as you can see here in the super resolution anoscopy images of Indian Moonchuck cells, control cells show this very characteristic, very thick K fiber, which is basically a fiber of microtubules that attach kinetochores. And these K fibers in Indian Moonchuck are usually very thick and very robust. However, if you now inhibit AUS6 or augmin, you no longer see these fibers here. So what Anna proposed at the time was that this protein was required for the formation of robust K fibers, and even more that it promoted the microtubule amplification from pre-existing kinetochore microtubules. And that is actually what you can see here in this experiment, which is a partial K fiber ablation. And so this also shows a very big advantage of using the Indian Moonchuck system to address these kind of questions, because given the fact that we have very few chromosomes and very thick and robust K fibers, we very easily can identify these K fibers here with the yellow rectangle, and we can use an ablation laser to specifically ablate this K fiber, as you can see here, and then follow the microtubule regrowth over time from the pre-existent kinetochore microtubule that was left there. And so this is what happens in control cells. However, if you are in a scenario where you don't have AUS6 or augmin present, you no longer see this regrowth over time. And this is actually what is quantified here as well, where you see if you have augmin after ablation, you are able to restore the microtubule based on the part of the microtubule that was left there after ablation. If you don't have augmin, then this recovery is much slower and less efficient. So this prompted Anna's model at the time, where she was able to find a kinetochore-driven augmin-mediated microtubule amplification process that was actually independent of pioneer centrosomal microtubules. I did not show you this last part, but you can always scan the paper here. And now, besides the Indian Moonchuck system that I was talking about in these two previous papers, we now started to use a very closely related deer species, which is the Chinese moonchuck. And the Chinese muntjac, as you can see here, in a diploid karyotype presents 46 very small chromosomes, just as in human cells. And these species actually prompted the evolution of the Indian muntjac species by a series of chromosome fusion events over 3 to 5 million years of evolution. And here is where we are collaborating with the Ruminant T2T Consortium, because this is a great chance to have access to telomere-to-telomere genomes from both species to be able to precisely identify the sites of chromosome fusion in the Indian muntjac chromosomes and eventually understand the mechanism behind chromosomal fusion. And so the main advantages of using these two systems in parallel is that they display a very similar genome that was kept during the chromosome fusion events, but they have very distinct chromosomal architecture. And besides having access to these two systems that I just presented you, I'm also able to work with tetraploid Indian muntjac cells that spontaneously occur in cell culture and that display 12 giant chromosomes. So I'm currently using these three systems in parallel of different Indian muntjac species, including the tetraploid Indian muntjac, to address two main questions. The first one being, how does the mitotic spindle adapt to alterations in chromosome number? And the second, how does the mitotic machinery itself adapt to these alterations? And so here we are focusing on three very important mitotic players, which are SNP, KIF89, and MPS1, that have been featured in recent papers that I'm showing you here, that state that chromosomally unstable tumor cells specifically require these mitotic genes for proliferation. Besides that, KIF89 and MPS1 are currently involved in clinical trials of phase 1 and phase 2, where chemical inhibitors for these proteins are being tested as anti-cancer therapy. However, the biology behind it remains unclear, meaning that the reason why cancer cells are selectively sensitive to these inhibitors is still unclear. And so our goal is to try to understand if differences in KIF89, MPS1, and SNP dependency might be exclusively due to alterations in chromosome number using these three systems that I just showed you, rather than other features that are commonly present in cancer cells, such as chromosomal instability. And so this is our lab. And the muntjac team is growing and growing nowadays. We are 5 people working with this system. Thank you all for listening, and I would love to take your questions.
Thanks, Inez. Great talk. Again, we'll have the questions after we wrap up with Janice.
Are you able to see my screen? Okay. Hi, everyone. I'm Jenna, and I'm very excited to talk to you today about my work during my PhD, where I customized deep variant for domesticated animals to improve our ability to discover new variation. And today, my primary goal is to give into one key point that deep variant is superior to GA to K. However, there are consequences when you use a variant caller that has been exclusively trained with human genomes. And I also plan to address the most common question that I get, what about my favorite species? And then I also want to leave you with this idea that we're working towards of a golden generalizable model. For any Lord of the Rings fans, you will get the reference of one model to rule them all. And this process was quite long and arduous, so we had to go on a bit of a journey to get here. And within that Lord of the Rings vein, I wanted to introduce the cast of characters. So in my world, we're working primarily with domesticated species, both internally and with collaborators. And the key challenge for us is that we're not working with humans. But for me personally, I'm interested in how can we use domesticated animals to improve our ability to identify variation in smaller populations such as wild or endangered species? And the challenge whenever you're trying to work with these wild populations is that you have a small sample size. And with our traditional approach, GA to K, there is this known limitation, specifically with VQSR or variant quality filtering, that this process does not work well when you have a small data set. But furthermore, GA to K is specifically designed for human genomes. So the best practices often get customized or tweaked. There's not a lot of consistency between labs working in the same species or even, across species. So there's varying degrees of effort, but also varying degrees of success at producing a high quality variation data set with these tools. So we know that the defaults are not optimal, or the defaults are optimal for humans, but they are not optimal in other species. So we see this when we look in our cattle and outgroup species data. So different VQSR parameters produce 60 million more high quality variants simply by changing how we run this algorithm. So when we first heard about deep variant, we were very excited. And again, as part of the Lord of the Rings, GA to K is more of the safe and familiar Shire, whereas deep variant is the shiny new Rivendell. So we were very excited that we would potentially have an alternative way to do variant calling that maybe would be less work. And our initial assumption was that this human-trained deep variant would perform similarly in other species. And we quickly learned that that was not the case. But given that we know deep variant doesn't work as well in other species, does that mean we are going to have to retrain a deep variant model for every single species that we work with? And with that, I'd like to propose, or what we're starting to propose, this idea of a golden generalizable model that works well in multiple species. And today I'm mostly going to be talking about cattle and outgroup species, but I just want to reiterate that we are using these species as a roadmap to do this work in any other species that we're interested in. And before I get into my results, I just want to briefly go over what makes a good deep variant model. So the primary metric that we use is called F1 score, which is simply the balance between precision and recall, and a value of 1 would indicate that a model is perfectly predicting your truth labels. And in humans, assessing models is relatively straightforward because they have a standardized truth set from the Genome in a Bottle Consortium. And no other species has these standard truth sets, so we're sort of having to have a workaround. And for this, we are using our high quality cohort variation data set that we created with GA2K. And because we used GA2K, there's some flaws in our definition of truth that are important caveats for how we interpret our results. And so to add an additional metric to allow us to compare performance across species, we use Mendelian inheritance errors, otherwise known as Mendelian discordancy. And I promised that, or I said that, deep variant does not work as well in cattle. And here is, you can see the difference between species. So on the top 2 plots, you can see the versions of deep variant that exist that were trained in humans and applying them back to the Genome in a Bottle human trios. And so their F1 score is quite robust. It's above 0.99, so nearly perfect. But when we take those exact same human models and apply them in cattle, we have much lower F1 score. So we're not doing nearly as well. And given this, the first question we asked was, can we do better if we retrain a deep variant model in cattle genomes? But before we could pursue this, we obviously wanted to make sure that our GA2K based approach for developing an alternative truth set was as good as we could make it. So for this, we had to go back and optimize VQSR to make it even better than the community standard. So for us, that's the 1000 bulls run 9 data. And what we ended up doing was tweaking the parameters, like I mentioned before, to maximize the number of high quality variants. So we're getting roughly half the number of Mendelian inheritance errors simply by changing our parameters. And in doing this, we realized that maybe we could retrain deep variant using these trios, and that would also allow us to identify these remaining Mendelian inheritance errors and remove them, and thus remove further errors from our potential truth set. And with that, I built an automated approach that enables iterative retraining. So we call this trio train. And this begins with the existing version of deep variant that was the human trained model. And then we provide training data from one of the parents in the trio, and then we assess the model for its ability to genotype accurately in the offspring. And then we select a new model, which becomes the starting point, and then we repeat it with the second parent in the trio, and it repeats over all of the trios that we give it. And as we are building these new models, we are also testing the new models in a separate set of cattle data and comparing that against our variant calls that we get from GA2K to sort of track how we're doing across this training period. And I just want to reiterate that the way we built Trio Train is to be species agnostic because we work in so many other species. So if you have the data, then Trio Train should work. And because we had multiple trios available, we were able to be strategic in how we built or designed our training process. So the first 2 phases are more of the typical approach or a cattle-specific version of deep variant. And then in the second or the third and fourth phases, we started to ask what would happen if we started building this multi-species deep variant. So we extended with bison trios during phase 3, and then phase 4, we use these F1 hybrids or these interspecies crosses. So I just want to also note that what primarily changes between these training phases is these variant distributions. So as we get into bison and the hybrids, we have changes to these distributions that deep variant is learning from. And so over the first 2 training phases, we primarily learn that the trios, which were from non-reference cattle breeds, so the ones that were not Herefords, actually performed better. So our theory is that they provide more homozygous alternate genotypes, and those are usually easier to call, and so it boosts performance slightly. However, we also notice this dip in indels on certain iterations that are reference breeds. So our expectation is that this would happen simply because of how we are defining truth sets, or how we are defining truth based on GATK. Lastly, in the third and fourth phases, we learn that it's not really possible to sort of game the system. So if you're trying to, say, improve performance on a particular variant class, that providing more of the data from that type of variation does not necessarily translate to a model that does better in, say, homozygous alternates. So when we train with bison genomes, we're increasing the number of homozygous alternate genotypes, but we actually have a lower performance. And then when we switch to these highly heterogeneous F1 hybrids, we actually improve performance because we are giving it so much more heterozygous genotypes. Then for comparing across phases or across these training strategies, we are just looking at this final model from each phase. And here you can contrast the 4 different phases against the existing deep variant in cattle. So at the top, you can see the 3 versions of deep variant. Again, the human trained models applied to cattle. And when we take our cattle trained models and apply them back to cattle, we have a much better F1 score, especially with SNPs. And we do see quite a bit of improvement with indels, although not nearly to the level that we have with SNPs, again, due to our definition of truth. But to sort of fully determine what's going on with our training process, we also wanted to assess Mendelian inheritance errors. So again, these are in F1 hybrids. And what you can see here is that the default version of deep variant. So if we hadn't bothered to retrain, we would have an order of magnitude more Mendelian inheritance errors than after training. And for anyone still on the fence about whether or not deep variant is superior, if we had not done any additional filtering with GATK, we would have had a substantially higher Mendelian inheritance error rate. So although deep variant out of the box does better or has fewer Mendelian inheritance errors, retraining was still able to improve it further. And what was very exciting for us was when we took our bovine models and applied them back into the human human genome antibody trios. And so here you can see the HG002 offspring that is the typical benchmarking genome. And what we see is half the number of Mendelian inheritance errors, even when we take our bovine model and use it in humans. So the reason this was so exciting was that we started believing that it was possible to create this species agnostic generalized model. And we still have more work to do, but it would be great to have a version of deep variant that can work across species. Oh, and I would also mention that we're reducing the number of Mendelian inheritance errors without performing joint calling. So we're able to get these data much quicker and still reduce inheritance errors. And I also just want to reiterate that deep variant is not aware of the species. That's not something it is able to detect, but what it is detecting is, again, these distributional differences between the training data relative to the human priors. And the reason we did all of this and why we spent so much time and effort is that we at the Mizzou Animal Genomics Group work in population. So the error reduction maybe in smaller sample sizes may not seem like a lot, but when you're working with, say, 9,000 cattle genomes, we're reducing our error by 300 and some odd million errors. So this definitely has the potential to drastically improve our ability to call variants. And then we can also use this model with in partnership with the T2T project, both using the assemblies as potential training data or using the long read data that was generated as part of the project to make different versions of deep variant. So it was very exciting that we had the ability to automate retraining process so that we can begin to explore these other questions. For me personally, my next steps are I want to be able to see if my cattle trained model will work well across the ruminant order. So in theory, or my hope is that my cattle model will work better than, say, the human model across species. And for anyone who wants to help, we are working on developing genome antibody resources in cattle. And again, we are doing this as a roadmap for other species. So with that, I will go ahead and take questions.
Thanks, Jen. So it looks like we do have a couple of questions in the chat. And more coming out. Great. So folks, if you want to unmute yourself and ask your question, you're welcome to. Otherwise, I can read them. So the first question's for Inez. Thanks for the great talk. Do you have any thoughts or evidence on the implications of centromere structure facilitating the fixation of complex karyotypes? Example, would you increase the functional centromere genetic core in Indian muntjac, an evolutionary response to counteract the strength of the muntjac inactive centromeres after telomeric fusions?
Okay, and thank you for all the questions. We would love to know this, of course. We still don't. We do know, so when you ask, would the increase of functional centromere kinetic counteract the strength? So we do know that the Indian moonchuck chromosome 3X, so the one that has the biggest kinetochore, is actually the result of these telomere-to-telomere or telomere-to-centromere fusions that then produce the compound kinetochore. So we know, and we have also seen in our lab, that we can find like it's repetitive because it's centromere, but like the centromere of the 3X is actually equivalent of multiple centromeres, one behind the other. So this would be an amazing system, and in particular, this compound kinetochore would be amazing to answer this question. We still haven't. So yeah, that's my question for now. But regarding then the second question, if we will be able to investigate other moonchuck species with intermediate or polymorphic karyotypes, the answer is I really hope so. So we actually established another collaboration within the T2T consortium with someone that is actually able to have access to these intermediate moonchuck species. So we really hope to get some karyotypes in the middle of the extremes, so in the middle of the Indian and the Chinese, to really understand if chromosome number is a player in the questions that we are addressing, for example, and many other questions that can come from there.
And then, yeah, there's a question for Jenna. Can you talk a bit more about the references used in training? I'm still a bit confused on how, if multiple references are used in terms of trio alignments for producing the training sets, or using a single reference in trio-related sets.
Great question. So we are using a single reference. So when I talked about training with bison or some of these interspecies crosses, we're aligning those back to the cattle reference. As far as expanding this to using multiple references, the new trajectory that we're working towards is treating these T2T assemblies as the truth set. So we can align those back to a reference and develop a known set of variation, and thus have a higher quality truth set. So that is rapidly developing and changing as the Q100 and the human T2T stuff sort of shifts the dynamic of how those truth sets, or our definition of truth, changes.
Another question, Jenna?
Oh,
yeah.
Thank you. Did you compare different error profiles from different sequencing platforms, and how robust are your new variant collars? So unfortunately, there's not a lot of long read data currently in our species. So we are just using Illumina sequencing and this population scale cohort. So we did not incorporate different sequencing platforms, but we would absolutely love to. And the plan is to use some of the new long read data that is being produced with the Ruminant T2T project in parallel as they're developing the assemblies, to sort of have a long read variant collar version ready to go when these assemblies are available too.
A long question, Jenna.
Okay. What is the source of the difference of performance in deep variant between humans and cattle? So that is a very nuanced and difficult question that I don't know that I can answer briefly. Primarily what we're seeing is that deep variant discovers a lot of new variation that obviously GATK misses. The challenge is going back and validating and figuring out what is believable and what is not. So there is a lot of particularly indels or repetitive regions that deep variant can identify, meaning the human version of the model, that we are unable to capture in our GATK truth set. However, if we were to just rely on the human version, there are potential genotyping errors. So it tends to skew more toward homozygous alternate genotypes, and misses the fact that we have a lot more heterozygosity in our species. So it's sort of a it's a nuanced answer or a nuanced question that requires a lot more manual work in order to validate, which is why we're moving toward developing an actual truth set for our species, where we will fully be able to quantify the differences between the models. And as far as your other question, why don't you send me an email? We could probably discuss further. That sounds like a longer discussion than maybe what we have time for.
Thanks. Jana, I actually had a question for you on the sort of origin of the ultra long D genes. And so just forgive my ignorance, but so all ruminants have D genes, right? And it's just that some of them are ultra long and others are just sort of normal length. And so you were talking about whether or not like sort of the gain or loss of ultra longs. But is there any evidence in terms of like looking at sort of duplication of D genes and whether some of these may have been ancestral and then lost? Or like, I guess the question is sort of like phylogeny of the D genes, if that would be able to help sort of tease apart when the ultra longs sort of were first emerged.
Yeah, this is a good question. And one detail that I missed on the talk is that we observed cysteine-rich D genes in many ruminant species. For now, I would say in all that we analyzed. So probably this is still beneficial to have such kind of antibodies, and just in some of them, they happen to be long. And if you look at the positions of ultra-long D genes in the IGH locus, and we also align them to other IGH loci without ultra-long D genes, we sometimes see that there are still D genes, but ultra-long D genes, they are located in this very small region with low complexity, because to have cysteine templates, you need to have the same nucleotides repeated over and over. So maybe that's the way these D genes were elongated, and in some species they just happen to be longer, and that was some sort of a beneficial adaptation, so they were less prone to purifying these repeats. But yeah, phylogenetic tree is a great idea, and we do see that there is a huge branch of genes where we see ultra-long D genes and short D genes, and they all cysteine-rich. And that's not just a ruminant feature. We also see cysteine-rich D genes in primates and in marsupials. They just not that tend to have longer sequences, and they have less cysteines. So that might be an adaptation, but that seems to be a mechanism that might be useful in all antibodies.
Cool, thanks. I know we're a little bit over, but are there any last questions? If not, thanks so much to the three speakers, and thanks for folks for joining. And if you're interested in the Ruminant T2T project, check out our manuscript and reach out to one of the working group leaders. Thanks so much. Bye.
Thank you all.
Thank you.
Thank you.